I'm really delighted to see you here. This is, I guess you might say, something of an experiment. It is something of a, this is a toy found in the bottom of the cereal box. I don't know quite how to describe it. It's something that we realize we should regularly do for members. And I really want to commend Charlotte Flemings for coming up with this idea and coming up with the particular presentation and discussion today with Rodolphe Barrangou. It's interesting the way science works, not always as predicted. Jeremy Berg and I were talking the other day about arguments that have occurred from time to time at NIH of whether it's appropriate for money from NIH to go to study, for example, plants or other non-medical, mammal-related science. For so many Americans, studies of bacteria or yeast should be just how to keep them out of your food. And in fact, they will be grateful when they realize what has come from study of how bacteria defend themselves. Who would be interested in preserving the health of a bacterium? How they do it turns out to be of great interest to us, as you will hear today. So I just wanted to say this is intended to be the first in a series of, what would I say, rewards to members of AAAS. I'm sure most of you don't know the half of what we do at AAAS. I find almost no one who does, but we do many things on your behalf. But probably not enough things with you and for you, directly for you, as opposed to abstractly for you. So when I came a few years ago, I pledged to do more with and ask more of our members, to really engage in both directions. We're doing that. We're asking more of members by using you as resources for our Sciline program to help writers get the science right in their stories, working with the expertise that is so deep and wide in our membership to help our new epicenter bring evidence to bear on public issues. So we are indeed asking more of members, and we indeed want to give more to members. And I think this presentation that you will get today is valuable. So we're happy to give it to you. With that, I would like to ask Valda Vinson, Deputy Editor of the Science Journals, to please introduce the speaker. Thanks.
Okay, thank you, Raj. And I just have a few slides to use, so I'm going to use my incredible technical skills here to okay. Oh, but I want it to be a slideshow, right? So, in 2015, Science selected CRISPR as Breakthrough of the Year. Maybe we're a little late to the game, but still we did it. But what was interesting is the news story that year began, It was conceived after a yogurt company in 2007 identified an unexpected mechanism that its bacteria use to fight off viruses. That was the first line in the news report. The scientist behind that discovery is Rodolphe Barrangou. And the journal who published that discovery is Science. So we were there at the very beginning of the CRISPR game together. And so, Rodolphe, we're delighted to have you here, finally, to tell us about what's happened before and after 2007. So when we were planning this event, we didn't want much from a speaker, just someone who could speak to the promise of CRISPR in biomedicine, in biotechnology, in agriculture, and to present it to us in the context of societal implications, and perhaps most importantly, to convey just how exciting the basic science is behind CRISPR. And as we discussed how challenging it was to find the person who could do this, my colleague in the front row, Steve Mao, said, well, then we need Rodolphe. So we asked, and luckily he agreed. And I didn't know as much back then, but as I've since learned that Rodolphe really is hashtag everything CRISPR. And that is the hashtag of the CRISPR Journal, where he is editor-in-chief. And when I really thought we've got the right person was when I read a sentence he used in an interview that said, we ask the questions that it would behoove us all to address as scientists, as consumers, as individuals, as academics, as investors, as parents, and as patients. So we're trying nothing less than to address all those questions with our members. Since the 2007 science paper, Rodolphe has moved back to North Carolina State, where as a professor, he's published over 100 scientific papers, and his impact in the field has been recognized by very prestigious awards such as the Canada Gairdner Award and National Academy of Sciences Awards in molecular biology and in food and agricultural sciences. And this year, he was very deservedly elected to the National Academy. But perhaps nothing reveals his enthusiasm better than his license plate. And the words that he used to close that interview with the CRISPR Journal, keep calm and CRISPR on. So, Rodolphe, over to you.
All right, so you know what the ending is going to be in the end. Do keep calm and CRISPR on. But why is it so important? And why do I feel compelled to advise everyone, whether they're consumers, whether they're investors, whether they're parents, whether they're individuals, or whether they're patients, why CRISPR matters? And the reason we're here today, the inaugural opening of the Science Matters series, and hopefully it is destined to be from AAAS a very successful series over time through the membership and the products and the ability of the organization to connect with the members to discuss why science matters and the technology rationale and the impact of technologies on biology is the title that was chosen by the journal. And what I've done is taken the liberty to adapt that to some extent because science does matter. But I'm going to argue today and I'm going to illustrate to you and we'll discuss in the questions thereafter cocktail that will follow my presentation here, how CRISPR technologies, with an S, have already continued to and will undoubtedly transform society moving forward. Again, whether you're an individual, a patient, a consumer, an investor, a parent, or any combination there. So in a quick show of hands, who's heard about CRISPR thus far? Who has invested in a CRISPR company? And who has consumed a CRISPR product, a food product or a medication? All right, so we'll see after the 30 minutes of talk I have whether you can invest in the CRISPR company and or you're going to run to the store to consume some CRISPR products or run to the doctor to ask for a CRISPR prescription. So we already live in a CRISPR world. CRISPR has been democratized. As a scientific technology, over 100,000 different CRISPR constructs have been distributed to scientists across the globe in thousands of labs in now over 100 countries. So whether you're here in the center of the world, geopolitically and scientifically, or whether you're in South America or even 4 regions of sub-Saharan Africa, people, at least in academia, have the ability to readily, affordably, and quickly, if you order your plasmids before 3 p.m. Eastern Time, you can get them tomorrow almost any institution in the US at cost for $65. This is the world that we live in today. But sadly, science is not the challenge. And you will hear in the next few minutes of how in a very short amount of time, merely 5 years after the first proof of concept was provided, that CRISPR is a technology that enables scientists to easily, readily, and affordably alter DNA in any organism you want, anywhere you want, almost just like that. We've been able to take the CRISPR science, the basic underpinning of what CRISPR is and how it works, and it's being packaged into a technology. Not CRISPR, the biological phenomenon that we heard about, the immune system in bacteria, but rather CRISPR, the tool. And that tool has been applied already to generate products that are on the market, or that have pending market approval, or that will be tested in the clinic starting by the end of the year. This is Q3 2018 right now, by Q4 2018, we will have CRISPR-based clinical trials going on domestically. So the challenge may not be in translating science to tech to applications during the products, but rather the acceptance of that technology by the public and by the regulators and by the governments that control the dissemination and the distribution and the use of those technologies. And this is where the innovator's dilemma stands, and this is why here, with a diversified constitution of AAAS, there's academics, there's industry, there's government, and there's society. And some of us wear many different hats. Some of us are scientists and academics. Some of us have worked or work in the industry. You have made it. Some of us are in the government. And then some of us are consumers. Some of us are investors. So let's start with CRISPR. What is this CRISPR thing? What is it actually? And ironically, you will see that most people who use CRISPR don't even know what CRISPR actually stands for. It's a cumbersome acronym that stands for clustered, regularly interspaced, short, palindromic repeats. I'll just say CRISPR from here on. And this is what it looks like. It's mesmerizing. It's beautiful. It's nonsense gibberish for the civilians in the audience. But for those of you who are geneticists, who look at those every day, you see something amazing. If you don't, I'm going to help you a little bit. You should see some uppercasing. And if you're not colorblind, this is what you should see. This is the same thing. This is a cluster altogether in the genome, regularly interspaced with exquisite periodicity, short, it's about 3 turns of DNA helix, 30, 36 nucleotides, partially palindromic, you can see the reverse complementation here of G, T, 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 with quadruple AC, and repeats. This is a CRISPR. It's probably the first time you see a CRISPR in your life. Sadly, it may be the very last time you see a CRISPR in your life, but you've seen a CRISPR once in your life. This is what it looks like. Clustered, regularly interspaced, short palindromic repeats. So now that we know what it is, what does it actually do? So one of the reasons you're all here today, most of you healthy, regardless of age and gender and all the bad decisions you've made over the course of your enjoyable life, is that we have, as humans, a system called adaptive immunity. And if you remember your high school biology, it hinges on the protein-protein interaction between antibodies and antigenes, right? Vaccination, lock and key, antibodies and antigenes. This is what we have as humans. And bacteria, for the longest time, were not thought to have adaptive immunity. This is the one thing we discovered back 11 years ago, which in the CRISPR world was like 50 years, now it will be 100 years. And essentially what CRISPR provides in bacteria is adaptive immunity. The same concept. But rather than be protein-protein driven, it's nucleic acid driven. DNA encoded, RNA mediated, DNA targeted. And this short sequence right here is a vaccination card imprinted in the genome of bacteria, which in between repeats captures a piece of viral DNA. It's the bacterial equivalent of a device like this. Imagine this being the bacterial CRISPR, and what the bacterium is going to do is take a picture, take a Snapchat, take a Polaroid, depending on how old you are, of the invasive genetic element and capture it. And this particular mugshot is going to be used subsequently by the molecular machinery of the CRISPR-Cas system to recognize something that has the same phase shape, in this case genetic shape, recognize it specifically, and cut its DNA accordingly. That's the way it works. I'm not going to talk to you too much about the science behind it necessarily, but CRISPRs will be shown like this. This is the CRISPR array. Repeat, space, and repeat, space, and repeat, space, and repeat, space, and repeat. It can grow up to 587 bad decisions the bacterium has made. All the repeats are the same. They're the black diamonds. And all the vaccination events are captured by a unique color scheme. First bad decision, second bad decision, third bad decision, and so on and so forth. And the way it works is a virus comes in, it's a red virus with red DNA, and the CRISPR is going to take its Cas machinery and capture a picture, a genetic piece of DNA from the invasive element, and integrate it into the CRISPR locus at one end. A red piece of DNA from the red virus. This is the vaccination event. Conveniently and genetically, this is an iterative process, so we can tell over time when the vaccination occurred and the order in which vaccinations occurred, genetically establishing a record. This locus is subjected to the centrodermal biology. DNA becomes RNA, so we make CRISPR RNA, and the pre-CRISPR RNA gets processed into a collection of small CRISPR RNAs I'm going to call RNA guides. And those small CRISPR RNAs literally guide the CRISPR-associated machinery called Cas to do sequence-specific targeting and cleavage of DNA. It's the CRISPR system using the CRISPR to try to match the genetic record to a particular phase of interest. It's going to recognize its head and chop its head off. DNA encoded, RNA-mediated DNA targeting. Now, nature sometimes is like a cook. I'm a food scientist. I'll talk about that in a few minutes. And imagine having different cleavage devices in your kitchen, like a knife, a butcher's knife, maybe a razor to shave garlic, maybe a chainsaw or a blender, depending on how domesticated or not you may be and what parts of the world you may live in. And CRISPR-Cas systems in nature take all kinds of cleavage devices. You have exonucleases that target double-stranded DNA and shred one strand, like the Pac-Man of CRISPRs, exonucleated. You have some shredders like this in type 3s that target RNA. You have the infamous Cas9, which is like a molecular scalpel. It has the ability to generate and trigger very precisely, very accurately, a double-stranded DNA break that is blunt. You have Cas12, formerly known as CPF1, that generates sticky ends. And you have a Cas13 that just shreds RNA. You can target DNA, you can target RNA, sometimes you can target both, and you can cut it precisely or exonucleotically process it. Now, CRISPR is not new. There's a whole history of CRISPR. CRISPR was born back in 1987. It was observed by a group in Japan investigating the non-coding region upstream of the IAP gene. I mean, it took 15 years to get baptized in 2002. That's when the CRISPR acronym was coined. And then it wasn't until 2007, as aforementioned, that we showed that it was a DNA-encoded adaptive immune system. At a company that was interested in understanding how food bacteria that make yogurt have the ability to withstand phage attack and viral attack. And between 2007 and 2012, A number of groups showed the CRISPR-Cas system mechanism of action. How does it work genetically? How does it work biochemically as a DNA-encoded, RNA-mediated, DNA-targeting system? But the world of CRISPR and the world of science changed in 2012. My friend and colleague Jennifer Doudna and her friends showed that you could use this molecular scaffold and take the scientific knowledge that we know about it to turn it into a tool. It's turning science into technology, the power couple, if you will, of translating scientific knowledge into a tool. And she developed a portable device called Cas9, cleavage enzyme, the molecular scaffold, and showed you can readily direct it to cut any piece of DNA you want in vitro in a test tube, accurately, precisely, efficiently. So imagine you having the ability to cut anything you want. And within 6 months of that discovery, another colleague, Feng Zhang, together with George Church and others, showed that you can use this device to do what's called genome editing. So bear with me for a minute. Imagine this molecular scaffold being a razor. What happens when you shave yourself occasionally? You cut yourself. Much like you would cut a piece of DNA right here. And what happens when you cut yourself? You may scream, feel a little heavy-handed, and you're going to bleed. And after that, you're going to repair that. You're going to patch it. You're going to build a scar. And this is what happens genetically for genome editing. There's 2 primary cut DNA repair pathways that exist in nature. One is called non-homologous end joining. You have your 2 strands of DNA and you just patch them back together like dirt or tape or duct tape. Patching those back together. When you do that, you're going to get a scar. So genetically, when you use an HEJ, you get a scar. You get some single nucleotide variations or SNVs or SNPs, small insertion, small deletion. You add a few bases, you remove a few bases, you get a genetic scar at the site of cleavage. There's an alternative way to do it. Let's say if you're more vain, you're going to go to the surgeon. And surgically, you're going to give the surgeon a template. Maybe your other cheek, your other thigh, or whatever part of your body that we hear about that you may shave. And you're going to say, I would like you to repair this cut with this template. Or you may be vain enough to say, I would like you to repair this cut with this template from some famous, good-looking younger person. Please redistribute the fat content accordingly or whatever you may want to do. And scientists can do that in nature and provision the DNA repair template and engineer exactly what you want to do at the site of cleavage. You may add something, you may remove something, or you may rewrite your DNA sequence. And that's why it's called editing, and we have a few editors, the true editors in the audience today. Because genome editing is the ability to rewrite the DNA sequence. You can change the letters, you can add a letter, you can add a word, you can remove a word, you can correct a typo. This is what Feng showed. And this stuff is still last year. Right? Now, much like editors, we have all the tools we need, thanks to CRISPR, to edit any genome you want, any way you want, in any organism you want. You can add something, you can delete something, you can knock something out, right? This is like CRISPR 1.0. Or you can use deactivated versions of CRISPR scissors and just turn them into DNA binding proteins. And the ability with which Cas9 will bind DNA is such that it will stop an RNA polymerase in its tracks. So if you're a biochemist, you're very impressed. If you're not a biochemist, it means you can turn transcription off. You can turn the light off. Or you can tether Cas9 to an activator and turn transcription on. Or you can tether Cas9 to an effector domain of interest, like a base editor, and alter one base precisely, from a C to a T. You can tether DCAS9 to a fluorophore and do detection and imaging of DNA. You can visualize a piece of DNA as you see fit, and you can do that multiplexibly, so-called CRISPR rainbow with different colors. And then last but not least, you can fuse DCAS9 or DCAS12, or soon enough DCAS8, to acetyltransferases and methylases to alter the epigenetic state of a sequence of interest. So it means that as genetic editors, we now have the ability to alter the genome, the transcriptome, the epigenome. We can visualize, change, alter DNA in any organism we want, any way we want. And in the last 5 years, with the democratization of CRISPR, thousands of labs, actually tens of thousands of labs, have been able to use those tools to edit all those organisms. You've been to the zoo before. Is there a zoo in DC? Is it any good? Yeah. All right. And it's free. Well, it ought to be free, otherwise people won't go. Okay? So you can do that to viruses, bacteria, yeast, worms, flies, so on and so forth, all the way to non-human chimps. And that's what academics do in the lab. They study model organisms, whether they should or not, whether NIH should spend more money on humans or not. We'll talk about bacteria and viruses. We can discuss. And then this is what the real people do in the real world, where money is being made, not just spent. And in the food and ag world, people have been able to show you can use CRISPR to edit plants, to edit livestock. You can make CRISPR chicken, not CRISPR, right? Same with shrimp. Talk about tomatoes and white button mushroom and corn and wheat. And then at the very top of the value and scientific pyramid, arguably, we have medicine. And people have shown unequivocally and with great power, they can use genome editing for all kinds of medical applications. You can do xenotransplantations, humanize pigs. You can do gene therapies, you can do gene drives, you can do antivirals. You can engineer stem cells. We might be able even to engineer human embryo cells and engineer humans. We'll talk about that. Because now we have the ability to do it all. And in the most visually compelling, in my opinion, example, I present you the CRISPR butterfly. You've all seen a butterfly before, and what's unique about butterflies is their wing pattern. And much like the human iris, the wing pattern of a butterfly is unique across the world. This butterfly is like no other. It has a unique wing pattern. And what CRISPR can do is you can take cells from that butterfly and use CRISPR to target a gene called yellow. And I'll let you guess what the yellow gene does. It encodes the color yellow. And you can grow a butterfly, this is what this one is smaller, from that one, in which the yellow gene has been inactivated. It's been CRISPR. And you can grow a butterfly with the exact same wing pattern, take my word for it, and grow a clone that doesn't have yellow in it. Or you can do that with other colors and change the color of the butterfly. This stuff is so last year, 2014. If you can do that to a butterfly, I would argue imagination is the only limit to what you can do genetically with CRISPR. If you can do that, you can do anything. Any organism, any gene, any sequence, any edit, any way you want. Affordably, quickly, efficiently, and scalably. We now have perhaps the most significantly disruptive and powerful technology in genetics we've ever had. Yet, this is a 5-year-old. We are year 5 of CRISPR. And like any 5-year-old, this is the most promising 5-year-old we've ever seen. It's programmable, specific, transferable, efficient, precise, affordable, quick, multiplexable, and scalable. This is what in 5 years has gone so far. But much like any 5-year-old you've ever seen in your life, it's imperfect. Think of the first computer 5 years in, the first car 5 years in, the first plane 5 years in, the first TV monitor 5 years in. It has a long ways to go. And there's things we need to do. We need to make it leaner. Like a 5-year-old needs to thin out a little bit, a little chubby right now. Cas9 is a little fat. Throws a tantrum here and there. We can talk about on and off targeting. It's predictable, but sometimes not so predictable. We try to control it and direct it, but we don't have full control over its specificity and efficiency, and more importantly, predictability of the genetic outcome. But this is just year 5. There's 100,000 scientists working on those problems and making CRISPR guides better and leaner and more efficient. And as always, right, we're going to have to figure out and control biology. Can we control DNA repair? Can we do HDR when we want to do it, as opposed to nature patching itself with a big scar? Can we predictably control the outcome of an egg? And critically, and we know that from centuries of pharmacology and research, it's hard to deliver drugs. It is still hard to deliver drugs. So think of diseases that are localized, whether in your eye, or in your blood, or in your muscle, or in your kidney. You have still to overcome the challenge of delivering efficiently enough CRISPR payload to the target tissue. We can't take a patient and dunk them into the lab, into a pool of CRISPR, and say, we'll be back tomorrow and see if it works. Okay, not here, we can't do that. All right. So accordingly, CRISPR has garnered the attention and the investments across different business segments, whether you're in research, whether you're in industrial biotechnology, bacteria, yeast, analogy, whether you're in ag, the big trifecta, plants, crops, animals for livestock, and bacteria are used across the food supply chain to make great products. Microbes are critical. Without microbes and bacteria, we have no yogurt, no cheese, no pickles, no champagne, no wine, no beer. Our lives would be miserable, even more than it is right now. And then last but not least, medical applications with almost no limit as to what you can do with CRISPR. Gene therapies, antivirals, cell therapies, immunotherapies, xenotransplantations, microbiome engineering, precision antimicrobials, gene drives, invasive species control. The list goes on and on and on and on. To give you a short examples of what we're doing right now in the lab, in bacteria oftentimes are a short-term predictor of what's going to happen next in more complex organisms. We use this vaccination card to track them and tag them. We use CRISPR as an antiviral, antibacteriophage, antibacteriophage, it used to be French, you'd say that. Plasmid control, uptake and dissemination of things like antimicrobial resistance genes in microbiome. We do genome editing, transitional control, large-scale genome editing. We can take out or sometimes alter hundreds of genes at a time. In bacteria, we're not there for humans yet, but we'll get there. And then do specific killing. And we can selectively target certain types of bacteria, the bad guys, as opposed to wipe out the whole microbiome, for instance. All right, so I asked a few minutes ago, who's ever consumed a CRISPR product? And only 2 of you, who know better than the rest of the audience, raise your hand. So let me walk you through this slide very quickly. This strain right here, DuPont Global Culture Collection strain number 7,710. I spent like 5 years of my life working on that bad boy. This guy makes the best yogurt in the world, just take my word for it. But unfortunately, this great strain is subjected to viral attack almost across the globe, whether you manufacture yogurt in the US, or in South America, or in France, or even Asia. There's all those viruses that prey on these bacteria. And these bacteria made 32 bad decisions in this life. And what you can do in the lab is take this guy, expose it to a bacterium, and let its immune system work naturally. And you select for a variant that picked up this piece of DNA and is now resistant. Take that guy, expose it to the second phase, become resistant, pick up a new piece of DNA, and so on and so forth. Within a couple of weeks, the average scientist in the average micro lab can use the natural version of CRISPR to vaccinate the yogurt starter culture. And ever since 2011, it's like 1 year before CRISPR, 1 BC. Since 2011, 100% of all starter cultures sold by DuPont globally to make yogurt or cheese have been CRISPR enhanced in the lab. This means that if you've had one cheeseburger, one nacho, one slice of pizza, one bite of cheese, one cup of yogurt, anywhere in the world since 2011, you've consumed a CRISPR manufactured using a starter culture that was enhanced with CRISPR. I'll let you think about that. And as a side note, this molecular record of events can be directed, and you can force the cells to pick up yellow, blue, pink, red, green tags. And if I see my yellow, blue, pink, red, green tag in your product, I know I made it. I know who's using it, I know who's buying it, I know who's using it industrially, and I can even tell who's eating it. We can now track bacteria all over the globe. And all those applications have been implemented, are being implemented, and will continue to be implemented across the food supply chain. Can we make cultures better for yogurt or cheese? Can we make probiotics better? Can we track things happening inside of your body, even when you're dead? Can we engineer bioprocessing, biomanufacturing workhorses? Can we control pathogens? And can we devise the next generation of microbes and bacteria used to make biotechnological products, biological products, and food products? The answer is yes. And you can now apply this across the food supply chain. For ag, better crops. For ag, better livestock. For ag, better microbes. For food products, like yogurt and cheese and pickles and sauerkraut and so on and so forth. And last but not least, for consumers, engineering your gut microbiome, your vaginal microbiome, maybe down the line your skin microbiome. We can do it all. So why are we here? Because this is arguably the most dogma-shifting scientific craze of our lifetime. CRISPR is everywhere. Not just in your food, but in the scientific news. You've seen those already today. Right? Almost every week, CRISPR makes the cover of the most famous scientific journals in the world. Very disruptive. Actually, if you look at history, science in particular stands out. Not just as the journal that took the first CRISPR paper back in the days when it was kind of exciting but mysterious and unknown. But in the CRISPR Olympics, science has the gold medal, the silver medal, and the bronze medal. And things like 5th place and 9th place and so on and so forth. Very compelling, very impactful, life-altering for some famous scientists. This is what I call the science craze, not science in general, but science journal. There's a lot of things that are very exciting that are happening right now. The power couple of science and technology is a reminder that you take your scientific knowledge, transfer it as a tool and a tech that you use and apply to generate products that will find their way to you as a patient or as a consumer, as an individual, not as a scientist, not as an investor, not as a curious member of AAAS. Right? And it's going to do things like change the life of genetic engineers, enable people to capture DNA codes and signatures over time to tell where things are going and where they come from, with unprecedented accuracy and speed and power. It is a revolutionary technology, and in this dedicated issue, we also see that now CRISPR is on the verge of being implemented in humans to cure genetic diseases. And we can target a faulty gene copy like DMD. Let's say you're missing exon 53, a correct version of exon 53, and we can come in, target it, cut it out, and replace it with a wild type copy, with a healthy copy, with a copy that is used by the large majority of humanity. We can restore your humanity using genome editing. And this is why it's been democratized so much. The numbers again, 10,000 constructs shipped as of last year. And look at the trends. Thousands of plasmids posited by hundreds of labs. And most interestingly is the papers. That first paper in 07, the whole literature was 27 papers. You could read the whole thing in one afternoon. And then he went to a paper a month, and then he went to a paper a week, and then he went to a paper a day, and now it's in excess of 10 papers every day. This quarter, the 10,000th CRISPR scientific publication was published from a handful just 5 years ago. It's mind-boggling. It's unbelievable. But the CRISPR craze extends beyond the scientific realm into the media. Not just the scientific media, but the real media, right? There's millions of hits on Google. There's famous scientists, part of the most influential people in the world. You can get your CRISPR news on NPR. You can get your CRISPR news on Twitter, #CRISPR. Every morning, maybe every even hour, you get the news. Never stops. #everything CRISPR. CRISPR made the cover of Time magazine. Not any issue of Time magazine, the July 4th issue of Time magazine. The most read issue of the year. As the editors note, it's not an accident. And as an avid reader of The Economist, this is the second time in the past 2 decades that science makes the cover of The Economist. It tells you everything you need to know. CRISPR is everywhere. But the real revolution is not just occurring in science. It's not just occurring in the media. It is occurring in the business. Because now we have people from pharma, from medicine, from food, from ag, and from biotech investing hundreds of millions of dollars in that technology. And they're doing this whether they're a Fortune 10 company like a GE, or a Fortune 500 company, or a private company, or a startup company, or even a not-for-profit company like an AG. Very rarely do you see one technology crossing the boundaries of business segments and business sizes and types. As a business school graduate, this is very compelling. And this is occurring with people like the top 10 biggest pharmaceutical companies in the world having officially placed CRISPR bets and getting access to very expensive and challenging and muddy CRISPR IP. This is why now we live in a CRISPR world, not just as patients, but maybe as investors. In the industry, the very top is the first generation CRISPR companies. Right? The Caribou's of the world, Intellia Therapeutics, CRISPR-TX, Editas. Most of them are publicly traded company now. As of the beginning of my talk, worth in excess of $5 billion today. Most of those companies became public within 2 years of inception of an idea to ringing the bell at the NASDAQ. That's crazy. And in the last 18 months, all those other companies have sprouted. On average, about one new CRISPR company becoming open every month. Right? Therapeutics, BMTX, Mammoth, PCBI, Genesis. Antimicrobials, Sniper, Eligo, Nextbiotics, Locust. Platforms, LifeEdit, Arbor, Cazymes, and Ag, Inari, and Pairwise Plant. One new CRISPR company startup every month. So with that scientific craze, and that media craze, and that business craze, and that investor craze, and a great technology come great responsibilities and challenges. This is the discussion we should have. There's little discussion to have about the science of CRISPR. There's lots of discussions to be had about the implications of CRISPR. Ethically, should we or should we not edit humans? Should we or should we not edit embryo? Should we or should we not alter the genetic content of humankind? people who are not even born yet, and even give consent. How's the public reacting to scientists saying limitation is your imagination? That's good, but for a lot of people, it's scary to think that scientists have no limitation. You're scared of scientists even with limitations, right? People are fearful, and the anti-GMO fear-mongering people are doing a very good job, sadly enough. How should we control and disseminate and regulate CRISPR? I went to Oxford to talk about the pros and cons of CRISPR in their official debate society. Tough crowd, educated crowd. The motion was, genetic engineering undermines the nature of humanity. And that motion was defeated after 3 hours of explaining why CRISPR is such a great tech. Convincing smart people is not that hard, because they're smart, they understand, they engage in the dialogue. They can look at the facts and the data and accept it. The same does not always apply to the science agnostics. And by the way, most scientists do a pretty mediocre job of explaining science to non-scientists. They even sometimes do a mediocre job of explaining science to scientists. And then how do we deal about corporations being so powerful? The Pope, His Holiness, convened a genome editing meeting at the Vatican. This is an actual picture from the Pope talking about CRISPR to a genome editing audience, arguing that we have a duty to care and a duty to cure. And I would argue, when it comes to the stewardship of humanity, I think the Pope opinion matters. At least we ought to listen to it. If we have a technology that enables us to cure human disease and alleviate pain, are we not ethically compelled to use it? I ask the question, I give not the answer. And then last but not least is how are we going to regulate this? Who should regulate this? How educated are the regulators? Who should do that? When should it be done and how should it be done? And by the way, we have to do this globally. That's hard. It's not really possible. You can't even agree sometimes domestically. So I had an epiphany. According to UNESCO, there's about 7 to 8 million research scientists in the world that went to grad school. There's like 7 or 8 million of us. And the population is about 8 to 9 billion. So for every one of the scientists, there's 1,000 of them. It means that every one of us who understands the science, can talk about the science, can rationalize the science, can discuss the science, right? We have to go out in the real world and convince 1,000 people about the benefits of CRISPR. And when I talk to my friends about this, oh my, tens of thousands of CRISPR friends about this, we are not doing our job. It's hard. It's difficult. Most people are not equipped to do it. Some people don't even want to do it. So one of the things we're doing right now is we're going to famous movie makers to make a documentary to explain science, to explain why science matters, to explain the great power couple of science and technology, to explain how CRISPR is altering your life, whether you're a patient or a scientist or an investor or a parent or a consumer or any combination thereof. And we'll see how we do. We will screen that in due time at AAAS and see if it's ready for prime time or not. And here lies the challenge of taking great CRISPR science, circa 2007 and maybe before, and turning that into a molecular scaffold to do genome editing, the technology, and using that genome editing technology and apply it to food, to ag, to biotech, to medicine, to gene therapies, to immunotherapies, to antivirals, to genetic diseases like Duchenne muscular dystrophy, But the limiting factor will be key opinion leaders, NGOs, journals like Science, societies like the AAAS, to help advance the science and the technology past this stage into acceptance by the public and consumers and stakeholders and the media. And this is what we're here to discuss now. So I'm going to thank the hosts for organizing this Science Matters series and inviting me to kick it off. And there's no better, more timely, or easier topic maybe to discuss than CRISPR. Right? Hashtag everything CRISPR at CRISPR Lab. As an employee, a state employee of the great state of North Carolina, I already disclosed my affiliation with a professor there, distinguished professor. I get funding from the NC Biotech Center, the NCI Foundation. My former colleagues and colleagues from DuPont and BASF and Pioneer, and my academic colleagues from Duke and Berkeley, as well as funding from Elysium, the corporation. I must also disclose the fact that I'm a former director at DuPont. I still have DuPont shares that I won't sell until they split next year from Dow. I'm a shareholder, and I cannot give financial advice to the audience today. I'm a shareholder and former chairman of the board of Caribou Biosciences. I'm a shareholder and co-founder of Intel Therapeutics. I'm a shareholder and co-founder of Locus Biosciences. I'm the editor-in-chief of the CRISPR Journal, so I like everything CRISPR. And I'm an advisor to an ad company talking about CRISPR. So if you're curious about me or about us and what we do, we'll speak now and engage with the audience. Have a few drinks thereafter, or you can reach out at your convenience. Thank you.
My name is John Cohen. I'm a reporter with Science, and let's give it up for Rodolphe. I'm going to talk for a few minutes and take questions from the audience. My job is to translate complicated science into plain English, but Rodolphe is so very good at it that he would put me out of business if every scientist was as articulate and as insightful about how to reach an audience. I think he spoke so clearly that I hope that your questions can be clearly answered, and I'm sure they can. One thing that I think is most provocative to me about your presentation is, given the $5 billion that have been invested, there is no product yet that has actually taken the tool of CRISPR, as opposed to what you did at Danisco, where you exploited what the bacteria already were doing, but taking that tool and applied it. Where do you think the first product will come from? A little soothsaying, what is that product? What will it be?
So people are really focused on medicine. Gene therapies is on everybody's radar. The journals, the media, the investors, the $5 billion valuation of the 3 CRISPR gene therapy companies, CRISPR-TX, Intellia, and Editas. And we're going to go into human clinical trials now. So this is what people want to see and use. And curing diseases is a very easy product to sell to the public. But in terms of timeline and ease of implementation, it's difficult. It takes years. We haven't started clinical trials. It's going to take years to do
And
it.
most things fail.
Most things, CRISPR won't fail. But
Most
so most clinical trials do fail, and they take a lot of time. And the FDA opines, and people battle and whatnot. So it's going to take time to get there. So I actually don't think, and I don't believe, that the therapies are going to be the first product. Rather, I'm of the strong belief that ag is going to win the story. And within ag in particular, it's going to be plants. And there's 2 reasons for that. The first reason is that it's already been done. Scientists already have been able to use CRISPR-based technologies in academia and industry across different kinds of crops. You can think of the big row crops like corn and soy and wheat and others like that, as well as things like the white button mushroom or tomatoes to enhance some of their functional traits. So Zach Lippman, great guy at Cold Spring Harbor Lab, has been able to show you can actually alter some promoter sequences in tomato plants to increase the yield of tomatoes by multiple folds within the same plant. And it's changing the branching patterns. You have pictures of a plant with a couple of tomatoes and one with like 3 times the amount. Very easy to see. Or people at Penn State, also affiliated at Penn State, have been able to show you can knock out the gene responsible for browning in mushrooms and take the white button mushroom and prevent browning, extending the bioavailability and the shelf life and the quality and the food quality of the mushroom. And people are using those technologies, again, in corn and soy and wheat and others, to enhance yield and enhance drought resistance and be more sustainable and be more productive. And this is done. We're just waiting for regulatory approval, which we have in the US, thanks to our great Secretary of Ag, Sonny Perdue, a real ag guy, who has decided and opined and confirmed that CRISPR-edited crops are not subjected to GMO labeling and regulation.
And we will see something different in other countries.
And we have seen something fundamentally different from our friends in Europe. And that's the challenge. The challenge is not technological, the challenge is not scientific. The challenge is regulation and policy and acceptance by the public. And if we want to feed the world, right, with an expanding population, by 2050, the world is likely mostly going to starve. If we don't increase our sustainability of food, our food supply, we're not going to make it. I mean, already people are suffering from malnutrition and limited access to food.
But let's open it up to the audience. Just come to the microphone with your questions. I can talk to Rodolphe for 4 hours. I'd like to give everyone here a chance to ask questions you might have. And one of the things I think that is most interesting is how we're going to have products that might be available in one place and not another. And I think that could create real dilemmas for exactly what you're selling. Please identify yourself, keep your question short if you would.
Sure. My name is Abhi Senka. I work at the Science and Technology Policy Institute. I was wondering if you could comment on the intellectual property side of this. I know that there's a lot of literature currently about the Broad Institute at the University of California and their patent wars over CRISPR. And if you could talk a little bit about the implications of intellectual property for some of the advances in agriculture or medicine. Thank you.
Great. It's a very important topic. It's been very well chronicled, documented, discussed. So there's this epic battle, notably in the US, between the Broad Institute and University of California, Berkeley, Fawn and Jennifer and others, of who invented what and where and how and who should own the rights to this. You can look at the many very good published opinions on this. The bottom line is that this reflects the potential and value of the technology. And in ag in particular, a lot of those rights have been secured in what's called the IP patent pool for ag by companies like DuPont and Corteva, and it's being made available globally to all the ag industry. So if you want FTO, it's there to be had, it's there to be licensed, it's there to be worked out. The same has not occurred as of yet in the pharmaceutical industry, and I've mentioned the top 10 announced licensing that have been secured by some of those aforementioned big pharma companies. Some have gone with the Broad IP, others have gone with the UC Berkeley IP. It's complicated because what we know as applied in the US doesn't apply globally. So there's a lot of patents that have been issued to both sides in different regions of the world. I cannot really give legal advice, I sure shouldn't, or I'm not qualified to give any legal advice, but it is complicated. It is not fully worked out. It reflects the value of the technology, big time, and the potential thereof. And there are many paths to freedom to operate and licensing available by all those parties and sometimes a patent pool. So that hasn't slowed down innovation at all. It's just slowed down the launching and commercial launch of those products, but not the development itself. It's not hindering science. It's not slowing down innovation. It's not preventing investments. It's being worked out and will be worked out. A lot of smart people working on this.
Just to bring everyone up to speed, the battle really boils down to 2 papers in Science. One paper came out in June of 2012 and showed that you could do CRISPR in simple systems. And then in January of 2013, a paper appeared that showed you could do it in mammalian cells, which means humans, which means medicine for humans. So the battle is between the biggest, richest universities in this country. It's Harvard, Broad, University of California, and some European players over that division of mammalian versus everything else. So just a quick synopsis. Yes? Question?
Yes, I'm retired both as a scientist and as a naval intelligence officer, and I know a lot about the interface between those two things, but there's one word that you used tonight that I don't understand at all, and I would hope you could explain it a little bit, and that word is biohacking.
So, in the world we live in today, CRISPR technology is democratized enough that people can do CRISPR in their garage, essentially. As a civilian, you can purchase equipment online legally and build a high school biology type facility in any garage or storage place that you want and play with CRISPR and tinker with CRISPR. And we have people who are on Twitter discussing, documenting, archiving, chronicling, and even misguidedly encouraging people to do CRISPR experiments at home. That's biohacking. I would argue it's interesting and intriguing from a scientific standpoint. I would certainly opine that it's very dangerous from a public health standpoint. And some of those people have had very adverse events. There was a loss of life earlier this year from a biohacker. So, science should be done by scientists in regulated, confined, controlled, safe, reasonable, supervised environments, whether they're in industry or academia or the government, obviously. But the world that we live in, across the globe, not just in the US, people are, quote unquote, biohacking CRISPR in their garage to edit the genomes of all kinds of organisms, including themselves. That's what I was referring to in one of my slides.
I did a story for Science, and we did a video that's online. Rudy Janish, who's a researcher who first showed that CRISPR could work in mice, said to me that any idiot could do it. So I tried it, and I failed. My experiment didn't work. I did it side by side with an experienced CRISPR scientist. So it's not true that any idiot can do it. I proved that. Just to clarify, biohacking is also dangerous because you can make a lot of mistakes, and I did. I'm not good at pipetting. Yes?
Hello, my name is Sharon McCoy. I am not a scientist, but I'm very intrigued by CRISPR. So I have two somewhat related questions. When you change the genetic coding for an organism, is this change permanent in the sense that future generations, children continued on, would have that modified genetic code as part of theirs? And then also tied to that, because I was thinking somewhat back to your slide related to the butterfly, and having, not to be punny, but having a butterfly effect in the sense of once you've changed someone's or an organism's genetic code, has there been any analysis of what are some of the reverberating effects, whether good or bad?
So, to go back to your first question, right? So if you change the genetic content of organisms, is it inheritable? It kind of depends where you change it, how much you change it, when you change it, and what organism you do with it. So in bacteria, everything is inheritable. You do that very fast, you carry it on and whatnot. In patients right now, most of the applications lie into local, topical, organ-dependent, or somewhat limited delivery. So if you deliver CRISPR to the blood, or deliver CRISPR to the liver, or deliver CRISPR to a muscle for DMD, you're not going to impact what's called the germline. You're not going to impact the ability of the individual to alter the cells that would be passed on to the progeny. Now, it's different if you take CRISPR and edit an egg, because all the cell in the organism that will come out of that egg eventually will be edited. And then that individual, if they go to reproduce in due time, would pass that on to the next generation. So conceptually, the answer is yes. To date, the answer is no. And that's the challenge between altering specific disease-related tissues, specific disease-related cells and control the delivery to a contained environment and not pass that on and not even change the large majority of the cells of the patient. But again, in the biohacking world that we live in the being aware and mindful of a lot of the regions in the world where regulations are not ubiquitous and are not enforced necessarily. You know, there are people actually as we speak who are working on editing the human germline and are planning, in my opinion, you know, to bring that to fruition. And that would have the consequences of altering the next generation of individuals that would come from the edited material. So the answer is yes, but we're just not there yet. And then there's many different flavors of CRISPR. Some of them, again, do not impact the genome itself. Some are transcriptome relevant, some are transient, some are localized. So some of those do not have dramatic impacts, globally speaking, on organisms. Sometimes it's just in a specific time series and moment in time with a specific genotype that it moves on and disappears. But again, the whole range of applications is in play here. And a lot of people are spending millions of dollars understanding what the ramifications are of altering CRISPR. We talk about on-targeting, off-targeting, unintended consequences, specific versus not, efficient versus not. And this is why it's going to take time and a lot of money to get to the clinic, because as of today, we still don't fully understand nor control what all the consequences of certain CRISPR payloads therapeutically may be in whole organisms. That being said, there's probably tens of thousands of mice that have been bred to date, and most of them look very good and very healthy and function very well. So there's a lot of encouraging data, but this is why it's going to take time when it comes to humans. We're just not there yet.
I think the interesting thing when I speak with people in the field is how nuanced much of this is. Even when you come to the question of germline editing, which is what you're asking about, changing sperms and eggs so that future generations are altered, CRISPR to me is like every other scientific tool. It can be used for good or for harm. It's like an axe. You can chop down a tree or cut off someone's head. It depends what you want to do with it. You can alter a germline, theoretically, to take out a bad gene that's in a family that's causing disease for centuries in that family, hemophilia or something. You could do things like that, so it need not be seen negatively. And it's often portrayed as that economist cover you showed about enhancing people, which is a great fear that we're going to have parents who want to have a child who you CRISPR edit so that your child can run faster and win Olympic medals. You know, that's the kind of horror fear of people. But the same technology could edit a germline in a positive way. So I think that it's so nuanced that you have to really look at it on a case-by-case basis to assess whether you think it's right or wrong. That's my philosophy. Yes, please.
Sujata Imani. I'm a AAAS Science and Technology Policy Fellow at Department of Energy. Being at Department of Energy, I don't come from an agro background, but they're actually doing a lot of biofuels work. I see that CRISPR could actually, I agree that agro is definitely one of the areas that it would make probably the soonest or the most early impact. I wanted to ask more on the regulatory side. Where do you think the regulatory bodies maybe need to communicate more with the scientific bodies to understand the consequences? And where do you think maybe they are doing a good job? For example, is FDA doing a good job where USDA needs to do better or something like that? And which bodies, or do we need a new body to address these genetic technologies that are coming up?
So that's very important because, as I mentioned, right, the questions are not scientific, the questions about regulation, acceptance by the public. So unlike most of my colleagues, actually, I'm pretty pleased with the job FDA and USDA have done, and I don't say that because we're in DC, and I hope to be back home tomorrow, but I mean, I think really they're doing a good job. And I'm going to mention Secretary Sonny Perdue having been educated and having looked at the question and addressing it and opining on it and publishing formal opinions from the USDA on the matter. I also know that the FDA is really paying very close attention to the pending clinical trials and protocols that come in. Actually, a couple of months ago, we had a pause on one of those efforts underway by CRISPR-TX because there were questions that needed to be clarified by the FDA. So I think they know of CRISPR, they're monitoring CRISPR, they're educating themselves about CRISPR. We still have a ways to go, obviously, but this is where technology needs to be further characterized and developed. Now, when it comes to DOE and biofuels, and I think some of the questions there are a little bit different because the process and the products, right, are different and being used in different ways. So you can use CRISPR to alter bacteria and then use those bacteria in the confined industrial environment to make enzymes. And then those enzymes themselves, can be used to do biofuel, right? I mean, how much are we at risk health-wise, depending on how the bacterium was engineered to make, this recombinant protein? We've been doing this for decades. Biotechnology industry has been built for this. And it works very well, it's very efficient, and it's happening. This is just one of the tools that we use for the genesis of next generation enzymes, for biofuels, for healthcare, for medicine, for industrial and houseware and enzymes that we use in laundry, detergents and whatnot. It's happening and it's going very well, and regulators are giving it the attention that they should.
We're going to wrap up. One thing I just want to point out, based on what Rodolphe just said, is in China they already have gone into patients with CRISPR alterations. And you had mentioned it will happen in the U.S. in the next quarter. It's going to happen any day now, I think, the first clinical trials. So we'll see that soon. But China's been doing it for at least since, I think, January, February of last year. And there are no results yet. We don't know anything yet. And it's going to take quite a while, I think, before we see results from anything that are positive or negative. Because that's how clinical trials move. It moves very slowly. And things that we learn most quickly are things that go wrong, not things that go right. So with that, thank you. Valda would like to close out, but thank you all very much.
Okay, thank you. It would be great to keep on going, but Rodolphe will be out, and we don't want to cut too much into the reception time. So just wanted to let everyone know, first of all, that Rodolphe did a great job advertising the issue that had 4 reviews on sort of technologies, and CRISPR was one of them. And there are journals on a table just outside, so you're more than free. You're very free to take one of those and read all the reviews in there. And then I just wanted to close off by thanking Rodolphe again, thanking John. If we want to give them just one more round of applause. And then I also wanted to give a big thank you to 2 other people who worked very hard on this, and that is Charlotte, who's standing way at the back there, who and then Steve is the other editor who worked with me on this special issue, and he was there through the whole conception and realization of this event too. So thank you, Steve and Charlotte.